I'm Jaden Rankin from the University of Texas at Arlington, and this summer I conducted research on measuring the effects, or lack thereof, of toxins from two different families on the spotted bird grasshopper. Animals have a wide variety of diets, many of which include eating plants. Plants, in turn, have adapted defenses to deter this herbivory. One of the adaptations plants have against insect herbivory is utilizing secondary metabolites they create as toxins. For example, in the solanaceous family, which includes plants such as tomato or eggplant, chlorogenic acid, or CGA, is used to deter herbivory. Another plant family, the brassicas, which include plants such as cabbage or broccoli, utilize phenethyl isothiocyanate as a toxin for defense against consumption. Insects, in turn, have response mechanisms to these defenses, such as symbiont-mediated detoxification, where the gut microbiota within the insect helps break down toxins the insect consumes. In this study, we aim to address the effect of CGA and PEI on the spotted bird grasshopper, Schistocerca americana, by feeding the insect a toxin-containing diet. Through this research, we gain a broader understanding of how these animals interact with their environment. Our hypothesis is that these toxins will have a positive correlation with mortality and a negative correlation with measures of fitness in both toxins, only intensifying an effect with increased toxin consumption. To study this, diets containing toxin were given over a 14-day period, during which time measurements and data are collected, such as weight at start and end of experiment, day of death, if applicable, amount eaten in PEI, date of instar change, and weight at new instar. The results of this experiment are very different depending on which toxin the grasshoppers were fed. In Figure 1, number of deaths in the experiment is shown, and there was no significance in death against the treatment amount of CGA. The same goes for Figure 2, where the CGA treatment is plotted against the total weight difference of the grasshopper over the course of the experiment. For the PEI treatments, however, the results are significant across the board. There is an increase in mortality as the PEI treatment increases, shown in Figure 3. In Figure 4, there is an average decrease in total mass of 42 milligrams for every increase in percent of treatment. The amount of treatment eaten by grasshoppers also decreases with an increase in PEI concentration, as seen in Figure 5. To conclude, there was no significant effect at any treatment level in any measure of fitness in CGA. In PEI, there is a significant negative effect on some measures of fitness, and an increase in mortality at the 1% and 5% levels. However, 5% mortality may be from starvation rather than toxin treatment consumption. Future research will use data collected from this experiment to test the hypothesis of symbiont-mediated detoxification in S. americana. Thank you.